Today, we are going to learn about protein drugs. Our objectives are to get ourselves introduced to proteins, to talk about a broad classification of drugs, to talk about advantages and disadvantages of proteins, and to talk about the classification and clinical use of protein drugs. And have an overview of the manufacture of protein drugs. So, what are proteins? So, proteins are basically, they're made up of a simple component that is repeated multiple times. And this simple component is the simple component are amino acids. These amino acids come in about 20 types. So 20 different amino acids repeated multiple times in varying sequences give rise to proteins. Okay. So if we have an amino acid joined to another amino acid, that in turn joined to another amino acid, and so on. Okay. So these amino acids, they join to each other by what is called a peptide bond. Okay. So when there are amino acids joined together by peptide bonds, we call it a peptide. Specifically, we are talking from 2 to 50 amino acids. So 2 to 50 amino acids make a peptide. So the simplest peptide would have 2 amino acids, because only 2 amino acids will be able to make a peptide bond. Okay. So based on the number of amino acids, and here each amino acid is called a residue. Called a residue. Each amino acid is called a residue. Each amino acid is called a residue. So now we have written this is a 4 residue peptide. Okay. 4 residue peptide. residue peptide. Now, depending on the number of amino acids, these peptides we again classify peptides. So these peptides, if it is 2 to 20 amino acids, if it is 2 to 20 amino acids, we call it a oligopeptide. If it is 20 to 50, amino acids, we call it a polypeptide. Okay. So then the question is, what is a protein? Okay, so technically it should contain greater than 50 amino acids. But that is not the only criteria. A protein should have primary, secondary, or a tertiary structure. Sometimes it may have a quaternary structure. So, what is primary structure? This, what we have written. Okay, so the peptide chain, peptide bond gives rise to a linear chain. And that is the primary structure. The secondary structure is when the linear chain folds. Okay, so when it folds, so these are all amino acids. This is an amino acid. There is interaction between the amino acids that folds the linear chain, and that gives rise to the secondary structure. The most important secondary structures are alpha helices and beta sheets. Okay. Alpha helices and beta sheets are secondary structures. Now, if this protein continue to fold and there is interaction from amino acids which are very far apart in the chain. Okay. Initially we had amino acid interactions between amino acids which are located nearby in the chain. But now if you have interactions from amino acids which are located far apart in the chain, then that gives rise to a tertiary structure. So up till now we are talking about one chain. Okay, one chain. If there are multiple chains, for example 2 alpha and 2 beta units like in hemoglobin, then there becomes interaction between the chains. Okay, so if there is, suppose this is chain 1 and somewhere there is a chain 2, and if there is an interaction between the two, okay, or more than or interaction with another chain here, okay, then it gives rise to what we call the quaternary structure. So let us actually look at what a real life protein looks like from its X-ray crystallographic structure. So let us transition to another software. Let me see. Okay, so let me pull up a protein. Which protein shall I pull up? Let's say I pull up protein which is 5Y VD. Okay, let's pull this up. Okay. Okay. So this is a protein. So this protein is called 5YVD, that is the description in the protein database. Okay, what is protein database? It is an online resource that contains the X-ray crystallographic structure of all the proteins which have been crystallized till date. So specifically, this is a non-structural protein. It is called NSP15 of the Middle East Respiratory Syndrome virus. Okay. So the virus that causes the Middle East Respiratory Syndrome, one of its non-structural protein is what we are showing here. It is called NSP15. Okay. So we have pulled up this structure to learn about the different components of the structure. So the line that I'm showing you now, this is a linear chain of amino acids. So this is the primary structure. This yellow structure here, which kind of is a helix, this is the secondary helical structure. The arrows that you see here, which are depicted here, this is a beta sheet. Okay. So anytime you look at a 3D structure in the ribbon format, you should now be able to identify the primary structure, the secondary structure. Okay, secondary structures, we said there is the alpha helix, and then there is these arrows, which are the beta sheets. Okay. So if you look at this structure again, you see there are two chains. Okay. So these two chains are interacting with each other. So basically this is the quaternary structure. And within one chain, we said the interactions, okay, it is the interactions that gives rise to this folded structure. And that is happening within a chain. So that is the tertiary structure. Okay, so we have talked about the primary structure, which is the linear chain of amino acids. We have talked about the secondary structure, which is basically alpha helix and this arrows, the beta sheet. And the interactions within the same chain gives rise to the tertiary structure. And when there is interaction between two or more chains, then that gives rise to the quaternary structure. Okay, so let us transition back to our software. Okay, so we now know the different structures. So why is this important? Because for a protein, the structure gives rise to its function. Meaning, if there is any change in this structure, then that will result in a change in its function. And that is why we need to know the structure of proteins. Okay. So with that, we have introduced what a protein is. Okay. So we have introduced what a protein is. Okay. Now let's look at broadly how we classify drugs. Drugs are broadly classified as small molecules, and large molecules. Okay, so drugs are classified as small molecules and large molecules. So what do we mean by a small molecule? It is defined on the basis of its molecular weight. So if it is less than 1000 Dalton, we call it a small molecule drug. If it is greater than 1000, then that is a large molecule drug. So proteins, we are studying proteins. Proteins are large molecule drugs. Okay. They are usually manufactured by recombinant DNA technology. They are usually manufactured by recombinant DNA technology. But there are also drugs which are not produced by recombinant DNA technology. So there are also natural protein drugs. Like what example shall we give? Papain. Okay. So papain is an enzyme from Carica papaya. Okay, the papaya fruit. So what is this papain? It is basically, it can degrade proteins, so it is a protease. So because it is large molecule, it cannot be given orally. Okay, so proteins, they cannot be given per oral. Okay, so then how can we give them? We can give them by the parenterally. Okay. So does any familiar protein come to your mind which is used in therapy? So if you said insulin, you are right. Okay, so how do we administer insulin? We give it subcutaneously. Okay. We also give it IV. Which preparation of insulin is given IV? If you said regular insulin, you are correct. Okay, only regular insulin is given IV. All other types of insulin are given by the subcutaneous route. Okay. So the question is, why are they given parenterally? Why can't you give it orally? If you give per orally, you know proteins are made up of amino acids, and in our stomach and intestine, not the stomach, the intestine has got proteases. These proteases can break down the peptide bonds. Okay, it can break this can break the peptide bonds. So if you break the peptide bonds, what do you get? You get lots of amino acids. So what did we say here? That the structure is required for function. Okay. So the protease, what does it do? It breaks down the structure. The structure is broken down by the protease. Okay, so the proteases convert the, say, insulin Okay, this insulin is converted to amino acids. So the structure of the insulin is lost. So if the structure is lost, it will not have function. And therefore, we don't give it orally. Okay, we don't give it orally. So we give it parenterally. So what happens if we give parenterally? So if we give parenterally, we have less activity of proteases. Proteases are less. So if there are less amount of proteases, what will happen? This structure will not be compromised. And if the structure is not compromised, we will get activity. Okay. All right. So that is the difference between proteins. So proteins we also call large molecules. We give different names. We call them biologicals. We also call them biopharmaceuticals. Okay. We also call them biologics. Okay. So there are different types of biologics. Proteins are a type of biologicals. Okay, it is a type of a biologic drug. And now we have seen what is the difference between large molecules and small molecules. Okay, the small molecules, they have got another interesting property. So small molecules, let's give an example, aspirin. Okay, aspirin acts on an enzyme COX. Okay, both COX-1 and COX-2. So what is COX? Cyclooxygenase. Okay, what does cyclooxygenase do? So cell membranes. So if this is the depiction of a cell membrane, the integral protein and a peripheral protein. Okay, so this is an integral protein. What is an integral protein? Which crosses both the layers of lipids. That is an integral protein, and this is the peripheral protein. So this cell membranes contain arachidonic acid. Okay, it actually contains phospholipids. Phospholipids under the action of phospholipase A2 gets converted to arachidonic acid. This arachidonic acid is acted upon by COX and LOX, cyclooxygenase and lipoxygenase, to the inflammatory mediators. Okay, so both give rise to inflammatory mediators. So our aspirin, what does it do? It blocks cyclooxygenase. So inflammatory mediators are not released, and therefore it becomes an anti-inflammatory drug. Okay, so it reduces fever, it reduces pain, it's an analgesic, antipyretic, and anti-inflammatory. Okay. So, a small molecule is supposed to act only on one receptor. Okay. This aspirin is supposed to act only on the COX inhibitor. But what happens is this aspirin, it also acts on the GIT, and what does it do? It reduces the formation of protective cyclooxygenase derived products. Okay. So this protective cyclooxygenase derived products, this protective cyclooxygenase derived products actually maintains the integrity of the mucosa. So they are mucosal protectants. Okay. So what happens? If you don't have mucosal protectants, then there will be formation of ulcer. Okay, so why are you giving aspirin? You got fever, you got pain, you got inflammation, and you have given this drug for reducing all that. But in giving this, it produces ulcer, which is actually not a desirable action. Okay, so what has happened here is that the drug acts on, it has got more than one action. More than one action. If it was acting only on the COX, it would have had only one action. Okay, but here we see so this COX is also again of two types. One type of COX, okay, so COX, there is COX1 and COX2. This COX2 increases in inflammation. Okay, this COX1 is a protectant. The protection that we were talking about is mediated by COX1. Now, we need aspirin to act only against COX2, which is produced in inflammation. Okay. But it also acts on COX1. So this drug has got more than one action, meaning it acts on more than one receptor. Okay. So this initial receptor, which is desired, okay, so this is the primary receptor. And this primary receptor is called the target. Okay. But it is also acting on COX1. This COX1 is called the off target. It's called off target. Okay, so there is a target and there is a off target. Anything that is not a target is called a off target. Okay, anything that is not a target is a off target. So what do we summarize from our discussion? That small molecules have off target effects. Because of off-target effects, the off-target effects are responsible for side effects. Side effects are caused due to off-target effects when the drug acts on more than one receptor. Okay, so in case of large molecules, okay, we are saying large molecules are more selective. Okay. So they have less off-target effects, meaning less side effects. Okay. They have less side effects. As the body produces many of these proteins, the immunogenic response due to the drug, the protein drug, is less. But it doesn't mean it is absent. Okay. Doesn't mean it is absent. Okay, it is less. So suppose a person has got a gene, okay, and this gene, suppose it is defective. Okay, and because the gene is defective, it is not producing the protein. Okay, or it is producing very less amount of protein. So then what happens? In this scenario, proteins can be used as a therapeutic intervention. Okay. So the other thing that you can do is you can do a gene therapy to solve the issue. Okay, but gene therapy has got its own problems. Okay, it has got its own disadvantages. So in that scenario, proteins is an option. Okay. The other advantage of biologics, of which proteins are a part, is that the approval time, okay, by the FDA, this is in the US. In India, it is the Director General, Drugs Controller General of India. Okay, drugs DCGI. The Drugs Controller General of India is the regulator. Controller General of India. Okay, DCG. I'm getting this wrong, sorry. DCGI. Okay, is responsible for approving. So, approval time for biologics is less than that of small molecules. Okay, so that's again an advantage of large molecule as a therapeutic agent. Then, usually proteins are unique. Okay, meaning they can be easily patented. Okay, what is the advantage of a patent? The company can monopolize. Only that company with the patent can manufacture the drug. Okay, so that is again an advantage. And then proteins as therapeutic agents, they are costlier. They are costly, okay, than when compared to small molecule drugs. So these are all advantages for the industry. So usually the approval time takes about 12 to 15 years for a small molecule drug. So 12 to 15 years for a small molecule. For a small molecule. Okay. So in this 12 to 15 years, the company is spending, the last count is 3.6 billion US dollars are spent by the company, and it takes 12 to 15 years to produce a small molecule drug. So if there is a decrease in approval time, it is an advantage for the company, because the company can start making money before 15 years. Okay. So if a molecule takes 15 years, only after 15 years they'll be able to sell and start making money. If it takes 7 years, then from the 7th year itself they can start making money. So it's an advantage for the company. Then patent. Okay, if you hold a patent life is 20 years. Okay. So the company holds a patent for 20 years, then it can exclusively manufacture the drug. So what happens after 20 years? Then if it is a small molecule, it becomes a generic drug. Okay, meaning many other companies can start manufacturing and it becomes generic. So if it is a biologic, after 20 years, biologic becomes a biosimilar. Okay, so now we have learned the difference between generic drug and biosimilar drug. We know now that the manufacture takes place 12 to 15 years and 3.6 billion US dollars for a small molecule. And the life of a patent is 20 years. After 20 years, the small molecule goes off patent and it becomes a generic. Okay. And biologic molecule, a protein, after its patent expiry, it is called a biosimilar. There are certain changes to the regulation. So nowadays, biosimilars can be produced by, there is the laws with regard to biosimilar is changing. Okay. So we have now covered the advantages of large molecules when we compare it to small molecule drugs. Okay. So which was one of our objectives. So we have talked about the advantages and we have talked about one disadvantage. Which disadvantage did we talk about? So if you have said parenterals, yes, it can be given only parenterally. Okay, so as far as the patient is concerned, it is associated with pain. Okay, and sometimes there can be local changes in the tissue. Changes in the tissue due to chronic administration. For example, insulin. Okay, you give insulin subcutaneously in one site for a long time, like in the abdomen, then it can cause lipodystrophy. Okay, lipodystrophy is changes in the lipids, so that area will become thick and hard. That is why sides are rotated. Okay, so it's not administered to the same site. Okay, we rotate these sides. Okay, then we talked about the economically it is costlier. Okay, costly when compared to small molecules. So those are some of the disadvantages of proteins as drugs. So the next topic that we're going to look at is the classification of drugs. Let me pull up a presentation here. Hold on, we will. There are a few other things that I wanted to talk to you about proteins. Let's take up as a question. Okay. What are the functions of proteins in the body? What are the functions of proteins in the body? So that's a question for us. Okay, so proteins play, they have got two roles basically. They play a structural role. So there are structural proteins and they have a functional. Okay, proteins have got a structural role and a functional role. And let's take some examples. Example of a structural protein would be tubulin. Okay. And functional proteins, what could functional proteins be? Functional proteins could be enzymes. Okay. They can be receptors. Okay, and receptors can be GPCRs. What are GPCRs? G protein coupled receptors. They can be ion channels. Okay. Or they can be transporters. Okay, so usually, so we are discussing what are the functions of proteins. So proteins have got a structural role and they can have a functional role. Now, tubulin, for instance, it is involved in what? It is involved in mitosis. Okay, so if you don't have tubulin, then the cell cannot undergo mitosis. Okay, or if the cell is dividing very fast, then maybe there is increased amounts of tubulin, like in cancer cells. So these structural proteins can also cause diseases. And functional proteins, we know if the enzyme is not working, then it can cause a disease. If there are problems with the receptors, either overactivation or underactivation, those can cause diseases. Transport proteins transport things into the cell and transport things out of the cell. If there is a problem with those, this can be a problem. Okay, so that is why protein drugs are important. Okay. The other question that we need to ask ourselves is, how are proteins produced in the body? Okay, how are proteins produced in the body? So the information molecule is DNA. Okay? This DNA is transcribed to RNA. Okay? And that we call, what do we call? Transcription. Okay? This RNA is translated to proteins. Okay, and this is called translation. After the protein gets translated, the protein undergoes modification. Okay, and those are called post-translational modifications. After the post-translation modification, it becomes a protein. Okay, and that protein, as we just discussed, it may have a structural or a functional role. It may have a structural or functional role. Okay, so this is the biosynthesis of proteins. Synthesis of proteins. Okay. So this is what happens naturally in the body. Okay, now I think we can pull up the presentation. So now we can talk about the classification of proteins. Okay. So how do we classify proteins? Is what we are going to learn now. So proteins, according to one of the latest classifications, it is classified into 4 groups. Okay. It is classified into 4 groups. Okay. So protein drugs are classified into 4 groups: group 1, group 2, group 3, and group 4. Okay, so what are group 1? Group 1 drugs are basically enzymes and regulatory proteins. Okay. So group 1 are enzymes and regulatory proteins. They are subdivided into 1A, 1B, and 1C. Let's see what they are. Let's take 1A. Okay. 1A are proteins that are used to replace a protein that is abnormal or deficient. Okay. So what examples come to mind? If you have said insulin, you're absolutely right. Okay. So in which disorder? Endocrine disorder. Okay, what is the disorder? Diabetes mellitus. Okay. And what can we do? We can here replace the protein. Which protein? Which is the protein that we use as a drug? It is insulin. Okay. So what is the problem with insulin? It has got a slow onset of action, and it has got a prolonged effect. Okay, slow onset of action. By that, what do we mean? About 30 minutes. Okay, and it's about 2 to 4 hours. Okay. So how do we administer the drug? We need to administer it at least about 30 minutes prior to food. Okay. If we don't do this, what will happen? can have early postprandial hyperglycemia. Okay. So what did we do? We did not give the drug 30 minutes early. So what will happen? The person has now taken food. Blood sugar is increasing, but the insulin has not reached blood. Therefore, the patient will have postprandial or early postprandial hyperglycemia. Then what can happen? Now the sugar has reached the blood and it is being consumed by all the cells of the body. So sugar is actually being taken up by the tissues of the body. But at this time what happens? There is increased amount of insulin reaching the blood. And that can cause late postprandial hypoglycemia. Okay. So that is why there was a need to modify the insulin. So in addition to insulin, what came? Insulin analogs. Okay. So usually in case of in the human body, physiologically, there is a basal insulin secretion. And then there is a bolus. Okay, so if it is, this is breakfast, this is lunch, and this time is dinner. So, in a normal person, before a person starts breakfast, what will happen? There is an increased amount of insulin. The x-axis shows increased concentration of insulin. So before breakfast, there is a spike in insulin. Okay. Before lunch, there is a spike in insulin. And before dinner, there is a spike in insulin. Okay, so this is called bolus. Okay. And irrespective of whether the person consumes food or not, some amount of insulin is always released, and that is called basal insulin. Okay, so there is basal insulin, there is bolus insulin. So with drugs, we are trying to mimic the physiological release of insulin. Okay, so here what is happening? Just before having food, there is an increased release of insulin, meaning if we are trying to administer a drug as an insulin, it should be given before food. Okay, it should be given before breakfast, before lunch, or before dinner. And for this, what is the criteria that we need? It should be slow acting. I mean, fast acting, fast onset. Okay. So this insulin should have a fast onset. Okay. And for basal insulin, it should be long acting. Okay, so to mimic physiological release of insulin, we need insulins which are having a fast onset and insulins which are long acting. So the fast onset analogs, what are the fast analogs that you remember? Yes, what goes in your mind? I hope you remember. If you have said it is Lispro, Aspart, or blue lysine, then you are absolutely right. Okay, so those are insulins which are having a fast onset of action. And what about long acting? If you said Detimir and Glargine, you are absolutely right. Okay. Okay. So the 1A drug which is given for an endocrine disorder. Okay, the first endocrine disease that we looked up is diabetes mellitus. Okay, and we have talked about different types of insulin. The other one which is given for an endocrine disorder would be, say, growth hormone. Okay, growth hormone. What is it called? Somatropin. Okay. There is also insulin-like growth factor 1. Okay, these are all given in patients who got less amount of growth hormone. So those are other examples. What other examples can we give? There can be GIT problems. Okay. Then we can give enzymes, proteases, which can be amylases. Okay. Lipase, other proteases. Okay, this can all be given. What about immunodeficiency? If you got immunodeficiency, which protein can you give? You can give adenosine deaminase. Okay, so it is required for the metabolism of adenosine. This adenosine deaminase metabolizes adenosine, and it is required for the immune system to function properly. Okay, what other examples can we give? What about some factors for coagulation? Let's give as a 5. So coagulation disorder, which disorder comes to your mind? Can be hemophilia. Okay. And what are the drugs that can be given for hemophilia? We have got factor 8, there is factor 9, factor 13, von Willebrand factor, etc. Okay, so these are again drugs that can be given for homeostasis. Okay. And can there be sixth one? What can we say? Some metabolic enzyme deficiency. Okay. And the drug would be alpha cerebrosidase. Okay, alpha glucocerebrosidase. Okay, so this is basically glucocerebroside. Okay, glucocerebroside. This is a lipid. Now, this lipid accumulates in certain genetic diseases like Gaucher's disease. Okay. That gives rise to the size of the liver and spleen increasing. People have pain. Okay. So, if this enzyme can be replaced, then it will degrade glucocerebroside to glucose and ceramide, I think. Yes, glucocerebroside. It will be converted to glucose and ceramide. Okay, and that is done by which enzyme? Glucocerebrosidase. Okay, so that could be another drug. Okay. So we have covered examples of 1A drugs. Let's look at 1B. What is 1B? So 1B are drugs which, let's take another color, that augment an existing pathway. Okay. With an example, it will become clear. These are usually drugs used in the fertility clinic for fertility. Okay, which can be follicle stimulating hormone, can be LH. Okay, so LH is usually lutropin alpha. Okay, and follicle stimulating hormone. So this can be given. Okay. The other examples would be for immunoregulation. Okay. Use interferons. Okay. So interferons can be interferon alpha 2A, it can be interferon beta, and beta can be 1A, 1B, 1C. Okay, all types of interferon. There is another interferon, pegylated interferons, PEG interferons. Okay, so there is a PEG interferon alpha 2A, there is a PEG interferon beta 1A, 1B and 1C. What is PEG? Polyethylene glycol. Okay, so it makes, polyethylene glycol makes the protein less immunogenic, more soluble. Okay, so there are lots of uses why PEG is used. So that is 1B drugs. So what about 1C? 1C, let's bring it here. 1C drugs. So 1C drugs are drugs that provide a novel function or activity. that provide a novel function or activity. And the examples could be Botox. Okay. Botox, and there could be hyaluronidase. Okay. What about papain? Okay. And for there can be streptokinase. Lepirudin, etc. Okay. So what is Botox? Botox is the toxin of Clostridium botulinum. Okay, it is the it's a neurotoxin. Okay. It does not allow the nerves to fire. So therefore it can be used for diseases called dystonias. What is a dystonia? Dystonia is a disease where there is excess contraction. Okay. So if you give Botox, this Botox will stop the nerve from firing, and therefore it can be used for dystonias. So hyaluronidase, this is actually used for drug delivery. Okay. Okay, hyaluronidase degrades hyaluronic acid, which are the cementing material of cells. So if you remove the cementing material, what happens? Absorption can be increased. Absorption can be increased. Then papain. What is papain? Papain, we said it is an enzyme. What enzyme? It is a protease from carica papaya. Okay, what is it used for? It can be, it's a protease. So it can be used for debriding tissues. Okay, so suppose there is a burn and the skin is in a very bad shape, then before we treat, we need to remove the dead skin. So we can do it mechanically, and if it is not possible mechanically, then this protease can be used to accomplish the same function. So streptokinase, it is a clot busting. Okay, it can remove the clot. Okay. So lepirudin, it's a what? Anticoagulant. Okay. Right. So those are the class 1 drugs. Okay. So what are class 2 drugs, group 2 drugs? Okay, so we have covered group 1. Now we'll come to group 2. Let's make some space somewhere. Let's talk about group 2 drugs. Okay. Group 2 drugs, we classify them as 2A. What is 2A? They interfere with a molecule. Okay, these are drugs which interfere with a molecule. And the examples of this class of drugs are monoclonal antibodies. Okay, which interfere with a molecule, and the examples are monoclonal antibodies. So let's discuss this thing in detail. So what is an antibody? An antibody is a protein component usually produced by a B cell, okay, and it is against an antigen. Okay, so it is basically a drug which acts on the immune system. It's a drug which is targeted at the immune system. So which is the best drug which is targeted at the immune system? The best drug is a vaccine. Okay? So antibodies are passive vaccines. Okay. So the best drug is not a passive vaccine, it is an active vaccine. Okay, so the best drug which is targeted at the immune system is an active vaccine. Okay. Why? What is the advantage of an active vaccine? There is memory. Okay. So once you immunize a person, there will be memory cells will be there which can mount an attack. And with each infection, the response is augmented. Okay, so with each response, with each infection, the magnitude of the immune response increases. Okay, and then more or less this is a permanent solution. Okay. But here we are using a passive drug. Okay. Okay. So we are using antibody which is passive. So then the question is, if an active vaccine is the best drug, why are you using the second best? Why are we using the passive vaccine? The reason is, suppose you are bitten by a snake. Okay. And the snake, the poisonous agent that the snake injects into the body is the venom. And this venom can be a neurotoxin, or it can be a hemotoxin. Okay, so you can have neurological problems or you can have a hemotoxin. Now, in this scenario, if you want to give an active vaccine, what would you do? You would take this venom, okay, and you would dilute it, and then you would administer it to your own body, and then wait for weeks and give booster doses, and you would become immune. Okay. But this takes time. Okay. This is a very slow process. So if you are injected by a snake which is venomous, you don't have this time. Okay, so if you try to do this, you would be dead. Okay, so then what is the option? You use the passive vaccine. Okay, so how do you do that? You would take this venom, you would inject it to an animal which has got lots of blood. Okay, why lots of blood? Because you're going to separate the antibody from the blood. So if there is more blood, it means you'll get more serum. Okay, serum contains the protein, the antibody. Therefore, if you need more doses, then you need an animal with more blood. So what would be your choice? It would be a horse. Okay, because if you want to make lots of antibody, if you want to do it in the lab, then your animal choice would be maybe a rabbit. Okay, but usually, commercially, the horse is preferred. Okay, so you dilute the venom, you inject it to the animal, and then you also add an adjuvant. Okay, what is an adjuvant? An adjuvant is something that will increase the immune response, increases the immune response. Because it is an animal, the adjuvant that you would use could be France adjuvant. Okay, France adjuvant is basically a preparation of mycobacterium in mineral oil. Okay, so this can be given along with the antigen. Okay, so the venom in small doses is the antigen and adjuvant. Is injected to the horse. Then we wait, check for the antibody titer. Okay, antibody titer. Okay, meaning we occasionally do tests to find out how much antibody is produced. Then we give a booster dose. And then we bleed the animal. And then we centrifugate, remove the plasma. Okay. Then inactivate the plasma. Okay. Plasma meaning you don't want any pathogen to be growing in that. And then you make it into a dosage form. Okay. So here, what happens is, let's take another color of the pen. Okay, what happens is, suppose, let's say, let me make my drawing a bit more easy to understand. So imagine this is the structure of snake venom. Okay. So we said B cells produce antibody. Now this antibody is specific to the antigen. This is the antigen. Okay, so one B cell will attach to this part of the venom and produce an antibody 1. Okay. The second B cell will produce an antibody against this. and the third one will produce an antibody against this part. Okay. So the different locations of the antigen is called epitopes. Okay, so the antibody is produced against different epitopes. And all these antibodies are present in this particular plasma. So this plasma contains all these types of antibodies. Okay. So this is called a polyclonal antibody. Okay, so what is the problem with this antibody? The problem with this antibody is batch to batch variation will be there. Batch to batch variation. There will be a horse to horse variation. Okay, so in some batch, AB2 will be greater. Okay. In some batch, AB1 will be greater. In some batch, AB3 will be greater. Okay. So you immunize one horse. In that horse, maybe AB3 is more than AB1 and AB2. The next horse will produce more AB1 compared to AB2 and AB3. So this is the problem. Okay. As a result, standardization of the vaccine becomes difficult. Okay. So that is where the next advance was, we don't use the traditional vaccine. Okay, so this is the traditional method. Traditional polyvalent vaccine. Okay. So the advance was, okay, this traditional polyvalent vaccine has got this problem. So how can we solve it? Okay. So how to solve? Okay, so the idea was, if you have a particular B cell which produces one antibody which you found very effective, say AB3, producing AB3, and if you can select this cell alone and grow this B cell to produce lots of AB3, then you have selected only one. So now you could call it a monoclonal. Okay, why monoclonal? Because it is producing only one type. Clonal, because you got it from one B cell which you tried to grow. So you took one B cell containing AB3 and you multiplied it. You grew it outside the body. So from one cell you got lots of identical cells. So this identical cells is called clone. Okay, so now you know the meaning of the word, how the word came about. Monoclonal antibody. Okay, so this was the idea, how to solve this problem. Okay. But idea and reality became different. Why? Because this B cell has got a problem. Okay. It will produce, it can produce antibody, okay, but it cannot grow indefinitely in vitro. Okay, because it is not a cancer cell. Okay, it is not an immortal cell. Okay, it can produce antibody, but it cannot grow indefinitely. Okay, at this time, people were studying myeloma. Okay, so what is myeloma? There are different types of WBCs. Okay, WBCs are of different types. One type of WBC is a plasma cell. Okay, so cancer of plasma cell is called myeloma. So people were studying myeloma and they found out the character of this cell. A myeloma cell can grows very fast. Okay, and it when we say it grows very fast, it divides very fast. It can undergo fast mitosis, and it grows indefinitely. Okay. So there were these two people called Milstein and Georges. Okay. So these two people thought, can we combine a B cell with a myeloma cell? Okay, so they are asking the question, can we combine a B cell with a myeloma cell? Okay, so if they could combine, then they would have a B cell which has got an antibody producing capability and myeloma cell which can grow indefinitely. Both properties they would have. Turns out they could do that. They could create a hybrid cell. Okay? Hybrid cell. Okay? So now you see how they got the name. Okay? A hybrid, and they use the word myeloma to come up with a new word. What was that word? Hybridoma. Okay, this hybrid cell, they called it a hybridoma. What was the advantage of this hybrid cell? It could produce antibody, okay, and it could grow indefinitely. Okay. So the technique used to produce a hybridoma, so basically what was being done was take these two cells and combine them in the presence of polyethylene glycol, and that results in a hybridoma cell which has got this property. It can produce the antibody and it can grow indefinitely. And so therefore this technology that was used to do this thing was called hybridoma technology. Okay. So these are monoclonal antibodies. Okay. So these are class group 2 drugs which interfere with the molecule. Okay. So group 2A drugs which interfere with the molecule, they are monoclonal antibodies. We have studied how a monoclonal antibody is produced, what is the difference between a traditional antibody, why a traditional antibody is called a polyvalent antibody, and what is the advantage of a monoclonal antibody. A monoclonal antibody, it won't have batch to batch variation, and we can control the type of antibody. Okay. So what are the examples of monoclonal antibodies is what we are going to look at now. Okay, are there what drugs can we make as monoclonal antibodies? So successfully they have been used for cancer therapy. Okay, the group 2 monoclonal antibodies. Monoclonal antibody. Okay, so that's the short form, monoclonal antibody. They have been used for cancer therapy. We are now looking at examples. Examples of monoclonal antibodies. So they are used for cancer therapy, and the drug is bevacizumab. Okay. It ends with a suffix mab. Suffix mab. And this mab means monoclonal antibody. Okay, bevacizumab. What does it do? We said it interferes with a molecule. Which molecule? It is an antibody directed against VEGF. What is VEGF? Vascular endothelial growth factor. Okay, so meaning this factor is required for angiogenesis. So this is the angiogenesis inhibitor. Then there is another cancer drug, cetuximab. This is a endothelial growth factor receptor inhibitor. Okay, endothelial growth factor receptor antibody. Okay, it's an antibody against endothelial growth factor receptor. Okay, again it is to do with the angiogenesis can be inhibited. There is another drug for immunoregulation. Which drug? This drug is infliximab. Again, it is a monoclonal antibody, and this time it is against TNF alpha. What is TNF? Tumor necrosis factor. TNF alpha. Now, TNF alpha, if it increases in the tissue, it will produce inflammation. Okay, so TNF alpha produces inflammation. If you can block TNF alpha, you can decrease inflammation. So it can be used in diseases like rheumatoid arthritis. Okay. So that is group 2A drug. There is also group 2B. Okay. So what is 2B drugs? These are drugs which deliver other compounds. Okay, drugs which deliver other compounds. Let's take an example. Let's take another color. What drug shall we take? There is a drug. Denyleukin diftitox. Diftitox. Okay. Denyleukin diftitox. So what it does is it delivers diphtheria toxin. It delivers diphtheria toxin to cells expressing interleukin 2. Okay. So it delivers diphtheria toxin to cells expressing IL-2. So what does this diphtheria toxin do? Diphtheria toxin inhibits protein synthesis. Okay. This IL-2 expressing cell is usually T cells. Okay, so normally, if T cells increase in number, then if we use this particular drug, we can reduce the number of T cells. Okay, so what does it do? This drug carries diphtheria toxin to T cells. As a result, T cells will not produce proteins. If it does not produce proteins, it cannot undergo mitosis. Okay. So this is given for conditions where there is increased T cell number, and that disease could be T cell lymphoma. Okay, so this drug, denyleukin diftitox, is a drug for T cell lymphoma, and that is an example of class 2B drug. Okay, group 2B drug. So we have talked about the second class of drugs. We have completed the group 2. Now let's come to group 3. What are group 3? Okay. What are group 3? Group 3 are protein vaccines. Protein vaccines. Okay, so let's take an example. Let's take example. Example could be HBsAg. Okay, what is HBsAg? It is a non-infective protein of hepatitis B. So this protein, if it is purified, then it can be used as a vaccine. Okay, so that is an example of group 3. It can be used for vaccination, active vaccination against hepatitis B. There is also the HPV vaccine. What is HPV human papilloma virus. Okay, so it causes cancer of the cervix. Okay. So, what is HPV vaccine? HPV vaccine is the capsid. What is capsid? Capsid is the protein coat of virus. Okay. So it is basically capsid proteins from 4 strains of HPV. Okay. Again, this capsid is not infective because it doesn't contain the nucleic acid. So you take the capsid, purify it, and then use it for vaccination. So that is an example of group 3. Then we come to the last class of drugs. What's the last class called? It is group 4. So what is group 4 drugs? Group 4 drugs are protein diagnostics. Protein diagnostic agents. Okay, let's take an example. The example PPD. Okay, what is PPD? Purified protein derivative, PPD. Okay. Purified protein derivative, which protein? Of Mycobacterium tuberculosis. Okay, so why is this used? It is used to test whether a person has got a previous infection from Mycobacterium tuberculosis. So basically, this PPD is an antigen. Okay. Okay. A person who has been infected with Mycobacterium tuberculosis in the past will have antibody. So what we do is we inject the PPD under the skin. If the person has previous infection, he will have an antibody. That antibody will react with this antigen and it will increase the, there will be like a wheal or a flare, okay, and on the skin. And if that is big, then we say that the person has had infection from Mycobacterium tuberculosis. So it is used as a diagnostic agent for tuberculosis infection. Okay, so that is the last class of protein drugs. Okay, let's pull up. I'm sorry, for some reason this is hanging. Okay. So up till now we were talking about the classification and clinical use of protein drugs. Yeah. So now let's have an overview of manufacture. So we said most of the protein drugs are produced by recombinant DNA technology. Okay. So what is recombinant DNA technology? Recombinant meaning you are combining. What are you combining? DNA from different sources which do not exist in nature. Okay, so that is basically recombinant DNA. The technology by which you try to cut and paste DNA from different sources is called recombinant DNA. How we do that is called recombinant DNA technology. Okay, so what it entails is basically we make a expression vector. What is an expression vector? It is basically a plasmid. Okay, so what are plasmids? They are non-replicating genetic material. Not non-replicating, they are non-chromosomal, sorry. Okay, so they are non-chromosomal genetic material. Non-chromosomal. Okay, so what is the property of the expression vector? The property of the expression vector is it can produce protein. Okay, so into this expression vector only we are putting the information. So suppose I want to make insulin, okay, A chain. I will take the DNA for the A chain, I'll put it into an expression vector, and then I will introduce this plasmid into a competent bacteria, and I will allow the bacteria to grow. After it grows, I will induce it. What is inducing? I will make it to produce the protein. So that will result in the bacteria producing insulin A chain. Then I will purify it to get the protein. Okay, so this stage, okay, this making an expression vector, meaning I have to first digest the vector with a restriction endonuclease. And I have to cut my protein information, my protein, my gene for my A chain. That also I will put extra sequence to that, which are restriction sites. So I'll put a restriction site here and restriction site there. And the vector will also, plasmid will also have restriction sites. Then I will use that same restriction enzyme, which will create overhangs. Okay, and then I will ligate it, and that will give me the complete expression vector. Okay, and then that I'll put it into the bacteria and grow it. Okay, so these aspects you have already studied as part of biotechnology. Okay, and I will make another lecture on how to produce recombinant proteins, but that is what I will leave for the another lecture. But here, all that I plan to do is to give an overview, okay, of what recombinant DNA technology is. Okay, so with that idea in mind, let us stop this lecture here.